The aim of the following experiment is to use voltage-sensitive dyes to optically record membrane potential changes and synaptic interactions between neurons in the central pattern generators of the crustacean stomatogastric ganglion. This is achieved by loading identified neurons with the dye Di8 and EPQ by iontophoretic injection using sharp microelectrodes and simultaneous extracellular recording of motor nerve activity. As a second step, the fluorescence of the voltage-sensitive dye is excited by green light, which allows the imaging of fluorescence changes that correspond to the changes in the cell membrane potential. Next, the image data is analyzed by selecting a region of interest in the neuron's membrane and comparing the optical signal to the recorded extracellular spike activity in order to identify action potentials and synaptic potentials in the imaged neurons. Results are obtained that show the optical recording of synaptically interacting pattern-generating neurons can supplement traditional recording techniques and open new opportunities for understanding how central pattern generator neural networks function.
The main advantage of this technique over existing methods like extra and intercellular recordings is that after dye filling, optical recordings of membrane potential changes and synaptic interactions can be achieved without the steric restrictions imposed by electrode placement and without further damage to the neurons during long-term recordings.
Though this method can provide additional insight into pattern generation in the crustacean stomatogastric ganglion, it can also be applied to other systems, such as locomotion and respiration in vertebrates.
Generally, individuals new to this method will struggle because dye selection